So please welcome Dr. Chauhan.
Thank you, Bob. Thank you, guys. I know you're all probably getting hungry. We'll try to do this fast. But it's going to be, I promise you, a fun and exciting, quick whirlwind tour of what's coming up next. So buckle up, because it's not going to be any standard Delta flight, but I'm going to be taking you in a time machine and show you a glimpse of what might actually the neuroendocrine world look like from years to come, and what is something that really gets me excited. So you all probably have heard from me, I am super excited about drug development, and that was one of the reasons I gravitated towards oncology, and I merged my passion for neuroendocrine cancer and drug development together to find help move neurotherapies and neuroendocrine cancer forward. You've heard a lot of key opinion leaders today about different aspects of neuroendocrine cancer management. Dr. Wolin spoke about latest trials. Dr. Vijayvargiya talked about targeted therapies. Dr. Muhammad talked about cool stuff in carcinoid world. So we have various tools, and Dr. White just gave an excellent presentation on liver-directed therapy. So there are so many tools. More tools, better it is. One of the tools that I would be talking about today is Theranostics tool. So what is Theranostics? This is one of those fields which is coming up big time. You've probably heard about it. If you haven't, then you'll hear all about it today, okay? So Theranostics is a concept where we use a small radio-labeled material, and it attaches to a cell-targeting agent to help us in both diagnosis as well as therapeutics. Let me give you an example to explain that. So you've heard about gallium-68 dotatate scan or copper-64 dotatate scan. So what are these scans? There's a radioactive gallium, which is attached to a somatostatin receptor analog. And by injecting this radioactive gallium, it goes wherever there is somatostatin receptors, which is primarily our tumors. And it lights up, gives small amount of radiation. And when we capture that on a PET CT, we know exactly where the tumors are. So this is the diagnostic component of Theranostics. Now, if a patient has somatostatin receptors, all we have to do is flip this gallium-68 and use a heavy-duty radioactive radionuclide like lutetium-177. Same concept. Now we inject that drug. It goes and sits wherever there is a somatostatin receptor, which is in the tumors. And instead of just radiating small amount of radiation to help us diagnose, this is going to radiate more of the tumor to help treat it. So a diagnostic tool becomes a therapeutic tool, so that's why this term Theranostics. Some people like to call it theragnostics, so diagnostic and therapeutics together. So very cool concept, and neuroendocrine cancer is where we've really spearheaded this, and we have lutetium-177 dotatate, first of its kind FDA-approved drug in the US. But there is another drug called lutetium-177 PSMA, which attacks PSMA, which is found in prostate cancer. And not only that, this concept is going to roll out in several other cancers. Look at this slide. We have breast cancer patients, pancreatic cancer patients, colorectal cancer patients. All we need is to find a suitable target. For example, this is FAP, fibroblast activating protein, seen in these cancers. So if you can tag FAP targeting moiety with, let's suppose, gallium-68, you can detect these tumors and later then treat them. So this field is going to really blow up in years to come. However, let's wind back and focus more on neuroendocrine cancers. That's what we care about, right? So this study, NETTER-1, phase 3 randomized controlled trial, done in about, I think, 14 countries, international study, large study, tested lutetium-177 dotatate and compared it to higher dose somatostatin analog for management of mid-gut neuroendocrine tumor. And what we see here is the median progression-free survival. That is, from the time patients got into the study and the time it took their scans to show progression, what was that median time? And look at those two curves. Patients who got Lutathera or lutetium-177 dotatate, they just did fantastic. Now we have enough follow-up data. We know this number is around about 29, 30 months. and the control arm did just 8.5 months. We just don't see these kind of results routinely in cancer world. And this was a huge success. And based on this, FDA granted approval. Now, hundreds of patients have access to this really transformative treatment in the United States. However, should we just stop there? Is it it? No. PRT is just now becoming mainstream. It's tip of iceberg. There are a lot of challenges that we have to tackle in next years and decades to come. I've listed some of those. Based on my preference, that's not the only challenges we have, but one of the things with Lutathera is, at least based on the FDA label of lutetium dotatate, the objective response rate is 14%. What that means is how many patients or what percentage of patients shows actual shrinkage of tumor. So it's a really good tool to stabilize the disease, but if we really need substantial shrinkage in heavy, high volume, bulky disease, our capability to do that with lutetium-177 dotatate is somewhat limited. So that is one thing that we should consider improving. The other improvement would be in median PFS. So 30 months, 29 months is very impressive, but how cool it would be if it can make it 60 months, 70 months, longer, right? So we should always strive to improve our current standard of care. And of course, there are other challenges that we have to kind of work on. So this is sort of roadmap that I envision as a theranostics physician scientist, that what we should be doing or what's currently happening in the field. So lutetium-177 dotatate is now a reality, which is a good start, but we should not stop there. There should be more and maybe better ways to treat or leverage our current theranostic agents and improve them further. And today we will be talking about some of those studies that are looking into those, okay? So one of the first studies I want to talk about, very close to my heart, is a trial using a drug called triapine. So triapine is a ribonuclide reductase inhibitor. This drug has a long history. It was originally discovered in a lab at Yale. I think we probably have to go 20, 25 years back. So it's a long track record of development. And what does it do? It inhibits a key enzyme which converts ribonuclides to deoxyribonuclides. What is deoxyribonuclides? Those are the building blocks of our DNA. So if we are able to hold off on that process, we actually help create an environment in the neuroendocrine tumor cells which prevents DNA repair after we treat patients with Lutathera, or at least that's our hypothesis. So this is basically an illustration describing conversion and how it works. So what got my attention on triapine? Triapine has been tested as a single agent in combination with chemotherapy, various trials, didn't really pan out, except for when they used it as a radiation sensitizer in gynecological cancers, and it was a huge success. This phase 2 study showed 80% metabolic CRs, complete responses, at 3-year time period. And this particular concept is right now in evaluation under registrational phase 3 trial in gynecological cancers, which is much aggressive cancer. But it really piqued our interest because at that time, we had an interesting radiation-based treatment introduced to us, Lutathera, FDA approved. So we thought, why don't we combine these two? Maybe we have a similar success in NETs. So we did some preclinical study. We never jump straight to human studies, right? We need to prove our concept in neuroendocrine tumor models. So we tested some of the pancreatic neuroendocrine tumor cells, BON and QGP. Those are the two models that we use at University of Kentucky, and they're also one of the commonly used. And as you can see the graph, the bar graph here, as we increase the dose of triapine in the cell culture, the cell death increased. All these cells are exposed to the same degree of radiation, 2 grays in this particular case. So we had some data, and then, of course, we later confirmed this in vivo or mouse models. We'll show some pictures later. It sounded like a resounding success in the lab, and we approached NCI to fund a first-in-human clinical trial to test the safety of this combination primarily, and also get early data on the effectiveness of this combination. So this study basically uses an oral triapine, which is a pill that we give with each cycle of Lutathera for 14 days. And we tested different doses up and down. We checked the blood levels, all sort of pharmacokinetics. And we also studied a lot of really good science. We didn't really just want to find a good combination, but also understand underlying mechanism of resistance, why some patients do better than others, and so many other things, newer biomarkers. So really loaded study. And you and all our taxpayer dollars are funding this federally funded study, so thank you. This is our phase 1 group from 2020 when we treated our first patient, and call it beginner's luck. Our first patient itself hit a partial response with 35% disease shrinkage. And I'm happy to announce that this study is now complete. And we are analyzing the data, and I'm looking forward to present the data next year in one of the big meetings, maybe AACR. And more importantly, this study is moving into a randomized phase 2, and the protocol is almost ready, and I promise you, I think another 3 or 4 months tops, we should have this study up and running. And this study would be accessible to all ETCTN EDOP sites. What that means is this study would be available at least 20 to 30 cancer centers all throughout the US. So you don't all have to come to Kentucky to get treated. We try to make it easy so that everybody can get access to this particular trial. So we will be randomizing patients between combination lutetium dotatate plus triapine versus the standard of care lutetium 177 dotatate, and we will see how well this works, okay? The other study that I'm very, very excited about, and this study actually started even before the triapine study, and it uses a very brand new drug. Unlike triapine, which was in the development for 30 years, piposertib is a very brand new, first in its class agent, which inhibits a particular enzyme called DNA PK. So when we give radiation-based treatments like Lutathera or chemotherapy drugs, essentially, most of the chemotherapy drugs as well, what they are trying to do is induce DNA damage. DNA is the engine of the cell. So if you damage the DNA, the cell doesn't function and it dies. Then you might ask me, Doc, once we get the chemo or Lutathera or radiation, then why does the cancer come back? Because the cancer cells are unfortunately also very smart. They have found ways how to repair their DNA. So this particular drug has been designed keeping in mind how to stop that repair process. And DNA PK is one of the key enzyme that we feel is, or we believe, that is involved in the DNA repair process. So this drug by itself doesn't do anything. If we don't pair it with a DNA damaging agent, it's as good as placebo. But imagine a scenario where we give a radiation-based drug, cause the DNA damage, and follow that through with a pill that blocks that DNA repair mechanism. That's a win-win situation, at least in theory. So this was my first extramural or federal grant, and I was a fresh off fellowship and starting my new job in July, and in August I received a package from Germany with all these new drugs. We developed these mouse models with neuroendocrine tumors. These are the subcutaneous, you can see them in the thigh. Those are the neuroendocrine, human pancreatic neuroendocrine cells growing in these the mice. And then what we did is we actually gavage fed. That means we mixed this drug, and then we fed them through oral route to the mouse, and then we radiated these tumors. And this is our first set of experimentation. So you can see from application of grants in February, and then getting the drug in August, and then we finished a year later in February. So it's a lot of work, but it was good work, and we were very happy to see some promising results. These are in vitro data which shows the cancer cells growing around Petri dish and culture plates. So higher the dose of piposertib, lower the colonies of these cancer cells. But I think you will appreciate the mouse data more because it's easy to understand here. So once we grew these mice, we divided them into 4 groups. One group of mice, unfortunately, got placebo, sugar pills. The other group got just piposertib. And as I mentioned, the M3814 is piposertib. It is as good as nothing because it really needs to be paired with a DNA damaging agent. So we sort of knew that would be the case. The third set of mice got XRT, the radiation, alone. And we saw there was some benefit, right? But the fourth group, the experimental group, when we combined piposertib, the M3814, with the radiation, look at the amount of tumor stagnation. It was just really, really good to see that. Not only that, we also observed these tumors were very avascular. That means, see, all these tumors are very reddish, and they are the combination arms very pale. So that means the angiogenesis was less. There was less blood vessels there. However, just an inside scoop. We finished all this in 2019, and the company, who is German Merck, and in the US, I think they're called EMD Serrano, they had their priorities on different cancers. So they shut down our project despite such good results. And it was truly a punch to the gut. But just like how all of you guys are net advocate, I'm also a net advocate. We did not leave any opportunity unturned. Every meeting we go, I would bother their employees. Hey, listen, what are you doing? Can you have more resources? Focus on NETs as well. I understand you have bigger cancers to worry about, but NETs is also very important to us. Eventually, after bothering them for a whole year, they proved the concept. They said, okay, we are good to go towards a human study. And I'm happy to tell you that this study is up and running. It's a multi-center study, and we are somewhere in the middle of it, and it's going to take some more time. But I'm very, very happy to at least take this concept from lab to our clinics and maybe potentially help some of our patients in near future. Just like our triapine study, we are doing a lot of cool science in this. We are trying to study tumor genetics, liquid biopsies, and a lot of other things. But very, very unique aspect of this study, we are also trying to study what's called dosimetry. Now, dosimetry is basically trying to figure out, are we giving enough treatment to each patient? One of the gripes that our nuclear medicine and radiation oncology colleagues have had, hey, you have this flat dose of Lutathera to all patients. How can one size fit all? And theoretically, they're right. There are pros and cons to this. It makes life easy for everybody, for patients. But they are also right in saying that maybe we are over-treating some, maybe we are under-treating some. Is there a possibility? So dosimetry is that tailoring of treatment to individual patients. So we are going to be doing first of its kind multi-center prospective dosimetry analysis in a cohort of this patient and understand, one, does it really help or just makes everybody's life difficult? Secondly, if it can be incorporated, what's an easy way to do it? Can we just get dosimetry at single time point and not really bother patients to come 24 hours, 48 hours, 6 days, 9 days period? So that's also a very interesting aspect of this study. Now, the latest Theranostics study that we've been dabbling our feet in is called Net Retreat study. How do you like the name, Net Retreat? So then the reason why we designed this study in collaboration with NCAN, so it's a cooperative group study. It'll be open, hopefully, in a lot of centers, maybe 100+ centers. That's our expectation, throughout US and Canada. So it's one of a very unique collaborative effort between Canada and the US. So CCTG in Canada, Dr. Simran Singh's leading that charge. And in the US and SWOG, I'm co-PIing this study. So Lutathera, we heard through NETTER-1 study, does very well. Patient have significant amount of stable disease. But after, let's suppose, 3 years, patient has now progression. The obvious question the patient would ask me is, Doc, it worked so well in the first go, let's try it again, right? The problem with that is we do not have prospective data to support it. The other problem is not FDA approved for retreatment, so the insurance might say, hey, we are not paying for it. So this study was designed to get some prospective data out to showcase that, yes, it can be done in a limited dose retreatment setting, and there might be safety and efficacy. So those questions are yet to be answered, but we believe that as long as patients fit, their blood count's good, their organ functions are good, and they had good, robust response at the first go with Lutathera, that we can retreat these patients safely, and they would hopefully do better than the control arm, which is everolimus, another standard care drug. So nobody's going to get placebo. You heard me on the day one, I'm not a big fan of placebos. So this is designed with that in the mind. And another interesting thing that we are evaluating in that study is biomarkers. Dr. Eric Liu gave a nice talk on biomarkers, and he also highlighted the fact that we don't really have good biomarkers. Chromogranin A is no good. And so we stumbled upon this particular biomarker recently called HPG80, and we looked about 91, 92 neuroendocrine cancer cases, and we actually found that this could be a pretty good diagnostic biomarker. We presented this data at eNets last year, and the whole pandemic spoiled the party. I was looking forward to an oral presentation there, but I had to give it virtually. But it looks very promising, and this particular net retreat randomized study will give us prospective opportunity to study how well can we just do this quick and easy blood test to monitor the disease. So down the road, can we cut down the frequency of CT scanning patients? That is our ultimate goal with this biomarker. So those were some of the things that we are personally doing and are excited about. I would also like to throw some lights on cool, cutting-edge Theranostics studies ongoing. A couple of interesting industry trials. For example, Compose and Compete. These are from a company called ITM, and they're looking at a drug called lutetium-177 idotriotide. Compose trial is looking at higher grade well-differentiated NETs because that is a critical unmet need. We don't really have good options there. And it's comparing PRT versus best supportive care, which could be one of the chemotherapy regimens. The Compete study is comparing the similar PRT drug versus best standard of care right now is Afinitor or everolimus. And we are eagerly awaiting these data because right now, for systemic therapies, for midgut NATs, we have 2 FDA-approved drugs, PRRT, and we have everolimus, but we don't know how to sequence them. So this particular study would be the first head-to-head sort of study looking at PRT versus mTOR inhibitor and might give us some glimpses of which might help us sequencing down the road. And NETTER-2 study is looking at Lutathera in higher grade well-differentiated NETs, and this study it's made public recently that has completed enrollment, and we are looking forward to its readout over the next few months or years. The other interesting NCTN study. So NCTN is a National Clinical Trials Group, again, federally funded groups. These are all paid by you and me through our taxpayer dollars. So very important studies to be done. And one of those is by Dr. Habde and Dr. Soares looking at role of lutetium-177 or Lutathera in pancreatic neuroendocrine tumor. So although Lutathera is FDA approved for both gastroenteropancreatic neuroendocrine tumors, but we don't really have prospective data for pancreatic NETs. And we also know from Dr. Kuhn's study in PNATs that CAPTEM is a very good option for our pancreatic NET patients. So which one should we go first? And this particular study might give us that answer. So it's randomizing patients between Lutathera and this chemo drug called CAPTEM. So very, very good study, and looking forward to seeing it complete enrollment. The other really important study to look forward to is role of PRRT in lung nets. On the day one, Dr. Robert Ramirez gave a wonderful lung nets talk, but he finished the talk by stating we don't have good options for progressive lung nets patients. especially the FDA-approved options. Besides SSA and Afinitor, there's nothing much. So this particular study would help us generate that data that might end up showing that there is role for PRRT in somatostatin receptor positive lung NETs patient, and compare to the best standard of care, best supportive care or standard, which is everolimus. And I would finish off with you've probably heard the buzz, the alpha PRRT. So lutetium-177 dotatate is primarily a beta form of radiation, ionizing radiation. There's another type of radiation called alpha radiation, which is a little bit higher energy. And there are now a couple of studies looking into. Okay, good, perfect. Are going to now finally investigate these alpha PRRT agents prospectively. Dr. Delpasan group through Alpha Medics is looking at 212-lead dotatate. They just published their early phase 1 data 2 months back, and it looks very promising. Just one of their patients, look at such nice response after 4 treatments. Of course, with the caveat, this is early data and needs to be now studied in a bigger phase 2 or a randomized phase 3 study, which is underway. The other interesting study, Alpha PRT study, is by Raise Bio, the Action 1 clinical trial, which is open at a lot of cancer centers throughout the US. So check them out. I've put in the NCT number there. And this particular study is looking at patients who have progressed on Lutathera. Again, an unmet need where they are looking at alpha PRT in those patients. So the future is bright, as you see, and hopefully in next years and decades to come, the whole landscape would have changed for better. So thank you so much. And this is my daughter and my dog. And these are my next projects. So thank you for your attention. It was lovely being here. And thanks, Marian, Bob, and Ken, for this opportunity.